So we'll obviously be taking questions. We have about 10 minutes. Could I start with one for Laura?
Okay, be gentle.
So obviously you're using all of these multi-omic approaches. Do you see very good concordance between the methodologies? So in your CyTOF versus your geomics?
We do seem to at the moment. Obviously, the spatial profiling is pretty new to us, but we certainly see concordance between our CyTOF and our proteomics.
Okay.
Yeah, so the team are unravelling that at the moment.
John.
Thanks very much. A question for Rachel, please. It is clear that not all IDH1 mutations partners are made equal, either IDH1 or IDH2. Are we putting our foot in it by designing our studies based on the fact that this is a very similar pathologies? Is methylation the bad guy here, and can we assume that?
So I think the assumption has always been that it's D2HG as a common mechanism across the mutations, and there's absolutely D2HG driven effects. So I'm hoping our models are going to try and unpick. So if we're able to fact sort out mutant versus wild type cells, what are is it a paracrine secreted mechanism? What are the cell autonomous mechanisms? So hopefully next time we speak, John, I'll be able to tell you, looking at those DMRs, those differentially methylated regions, what are the differences? Because I think the in vivo data is really critical here as well. So I've given quite a superficial overview, and they've got, obviously lists of genes for each mutation, but I think until we've got the mouse work down, I'm reluctant to go down a specific rabbit hole until we've sort of validated what's also true in the in vivo setting. It would also be extremely nice to look in human disease. So if anyone wants to collaborate on that front, it would be really useful, I think.
And then just to follow on from that, as we know, the drugs that we have don't work in a lot of people. They simply don't work. There's absolutely no benefit whatsoever. So is there any indication from any pathway stuff that you've done as to why it doesn't or does work in individuals?
I know Nabil's obviously done a lot of work in terms of the inhibitor emergence of IDH2 as an escape mechanism, but that's certainly not the whole story. And I think that's going to be one of the really exciting things that unfolds as we look through. So at the moment, not yet, but I'm hoping this platform we've set up is going to be really useful in terms of unpicking that.
Thank you.
So to kind of just ask a question on that. So I suppose there's this idea that cell state plasticity is quite an early hallmark of cancer, right? So transitional moments. How fixed does a cell become once it's changed its DNA and its methylation? So are the inhibitors able to shift it back to a normal phenotype or not?
Very good question, and I think as yet unknown. Certainly the evolution type studies in glioma have shown that IDH mutation occurrence is very early on, and the other drivers, particularly RAS, is a more late stage. Our data suggests that the metabolic environment of the liver, in particularly the periportal, glutamine transition, is a selection advantage for IDH. If you're IDH mutant, you're going to survive in that environment in a much, better way. But be very interesting to do those studies and add the inhibitor and reevaluate the metabolic and lineage effects of those cells.
Yeah.
Could I put a comment? Rachel, great. You mentioned about the anaprotic pathways. So it means cancer cells can use asparagine, glutamine to feed in the TCA cycles. If you lost the IDH, you might activate those pathways to feed it in, therefore leading to the maybe drug resistant. It's quite adaptable. So.
Yeah, I think that's very possible because our CRISPR system, show that the analyses we've done are very early after CRISPR, and I think display the acute effects of either D2HG or mutant IDH very nicely. So one would hope that, you know, if you inhibit that, you are then, you know, going to see some changes. But yeah, to be seen what.
Sorry,
So-
yes, oh, sorry, Sheila.
Oh, sorry, just to follow up, Rachel. So you are expecting to see quite different chromatin landscapes from each different type of mutational variation, I'm inferring. And so do you think that that's going to open up combination inhibition
Yeah.
for you for
So the HOXA.
treating mechanism?
The HOXA observation is very tantalising, and although we've looked at HOXA promoter methylation from our EPIC array, so there's hypomethylation of the HOXA promoter and therefore derepression of HOXA, the fact that that whole cluster is derepressed to me suggests there's other things going on, probably histone modifications. So some kind of a TAC-seq or other sequencing is definitely going to be required before I spend a lot of AMMF money characterizing that. I want to know, is this reproducible in the mouse? Is it reproducible in human cells? You know, before you spend your cash, you've got to be quite sort of have confidence in the rabbit hole you're going down. So absolutely there's going to be chromatin changes. So, you know, once we get those sorted cells out of the animals, I think that, you know, that will be one of the first things that we look at.
Okay, thank you.
Sorry, yes,
Yeah,
thanks.
I would like to discuss with Dr. Randall about your DSP experiment.
Okay.
First question is that did you set up a lower limit of cell number for each ROI? And the second question is that because you select only 2 ROI for each tumor samples, would you worry about the sampling error or sampling bias would be significant?
Thank you. With regards to low threshold, I'd have to defer to the team behind you as they've conducted the analysis. Yes, obviously we are very much cherry picking. So when we are collecting our specimen, we've done a lot of sampling and a lot of batch corrections actually. Obviously we've got a number of different TMAs and a number of different runs. So I know the team have been very mindful of that. We allowed the pathologist to identify the areas, the tumour cellularity on the sections that we've used. So the cores were taken from that. The selection biases, again, I'd have to defer to the team. They've done the analysis, so they're behind you. Wave, guys.
Could you answer or discuss about this?
Yes,
Owen, come to the microphone.
so for the lower cell threshold limit, we didn't want to use the DAPI stain for stroma because then it could exclude large fibroblasts. So we used it as not an exclusion factor, but as a flag for something that might have particularly low staining. We might want to review it because if it showed a poor result, we might have to exclude that region. So we actually normalized the surface area is the way that we did the analysis. And if there's a very low surface area and poor housekeeping genes, then we excluded the sample.
You did not count the cell number, for example, the low limits 100 or 200.
We have logged this as well. Yeah.
100.
I think, did we say it was 100?
And how about
Yeah, it was
the
100.
origin of the tumors? Do you use the tumor resected or biopsied? Because for the biopsy, the tumor sample is fragmented, and even the experienced pathologist, the selection of ROI will be difficult.
So this
and will introduce significant bias.
So this isn't a biopsy. This is the resected tissue.
The TMA come from resected tumor or biopsied tumor.
It's from the resected tumour, but this is FFP, so it'll be taken from the tumour block.
Usually we will select, especially you segment 2 areas of tumor. One is tumor center.
Yes,
and
we've
one
done
in
that.
Periphery.
Yeah.
Usually we will select at least maybe 3 ROI for each representative regions, but you will increase your cost expense.
Exactly. We did what we could with the money that we had.
Kevin.
Hi. I'll ask quick questions because I know it's lunchtime. So just first one is for Laura, which is,
hello.
you said that you have the tissue slices at the liquid air interface. So what is
Yeah.
the rationale for that? Because won't that mess up all of your oxidative levels and oxygen levels?
So actually we did some mathematical modelling originally. This is quite an old technique and a lot of people just allow the slice to float at the bottom of the well and then they try to ensure viability actually by putting it in really hyperoxic conditions. So the mathematical modelling actually demonstrated if we put our slices onto a membrane and insert and just put it just below the surface of the liquid, we maintained our viability. We had oxygenation levels, certainly on our liver cells, which were much more representative of the human situation actually within inside the liver. Obviously, I appreciate that a tumour cell is much more potentially hypoxic. So that is a caveat of our research, actually. Yeah.
Thank you. And then the second question for Shiji, which is, I didn't understand the rationale for doing a time course when you have the initial effect and then the effect disappears. But I mean, in a tumor situation, it's in equilibrium. So what is the rationale for the time course?
Thank you. Give me an opportunity to answer the question. But before that, I'm sorry, I have to say, first, I would like to acknowledge my team who work on this project, while Medina done so much on this study, and also including my peers, DPhil student, Gemma Owings, and also have a student from Konkan University also being involved in this project sometime last year. Now I come back to your question. So you know the metabolic kinetic change it won't stay stable. It just responds to the microenvironment. It will shift and change, adapt to the signaling cool to survive. So the reason we designed to look at the three sort of time points, and firstly, the technical is available. Secondly, our samples are available. Therefore, we just want to see the interaction between two cellular compartments, whether the shift of for example, glucose metabolism, amino acid metabolism would be consistent rather than catch one time point, you said up and down, but next time point it might be opposite. So that we'll get a consistent sort of be more reliable results to test our hypotheses.
Thank you both.
Thank you.
Well, on that note, I would like to thank the speakers again and the audience for your questions. And this is close to the session and lunchtime. So.
Excellent. Thank you very much.